Hi, here's Camila talking. I will make an introduction about breast cancer, which is characterized by uncontrolled growth of cells with genetic damage or mutations. The known risk factors are increase of estradiol due to early menarche, late menopause, and use of oral contraceptives, as much as age, family history, and obesity, which will be the focus of the discussion. On the other hand, childbearing, breastfeeding, and physical activity can reduce the risks and can even represent a protective effect. The main gene mutations known are BRCA1 and BRCA2. They are related to breast and ovarian cancer. P53, that is involved in predisposing to lean from any cancer syndrome, and P10, that is responsible for cold and disease. But there are many other genes associated with breast cancer that represent different kind of risks, as shown on the table below.
Hi guys, I'm Beatriz and I'm going to talk about the relation with food and dietary routine. There is another important risk factor in breast cancer physiology. There are some studies that show the direct relation between a high-fat diet and the increase of the risk of the disease, being about 50% higher in postmenopausal obese women with a body mass index over 30 when compared to lean women. And we can see how high and how expressive these numbers are. Besides this fact, the obesity is also related to higher risk of lymph node metastasis, large tumors, and death versus non-obese breast cancer patients. Some hypotheses proposed to explain these associations suggest that obesity in postmenopausal women elevate the circulation of hormones like estrogens and also insulin and insulin-like growth factors. These last two releasing metabolic syndrome, acting as a mitogenic compounds that can contribute to the tumor development.
Hi, my name is Giovanna and I'm gonna talk a little about the study chosen. After many searches at the GeoGene database, which contains more than 10,000 studies related to human breast cancer, we found a study called Gene Expression Profiles of Breast, Colorectal, Prostate, and Non-Small Cell Lung Cancer. This study holds the genetic profile of 280 samples of several solid tumors, of which 65 are from breast cancer. We thought this study was relevant because there were a lot of data that we could base our hypothesis on, like the existence of lots of information about the physical characteristics of the women with the tumor, such as weight, height, age, gender, body mass index, stage of the tumor, etc., and a considerable number of samples. So, to perform this analysis, we established two groups: one control group with women positive for breast cancer and an ideal body mass index, and a comparator group with women positive for breast cancer and overweight. With this, our question to this study was: is there a relationship between the development of breast cancer and overweight?
My name is Davi, and I'm going to show you some information that we acquired by performing the GeoGene analysis. So right here you can see that we sorted the genes by the log-fold change value, and we took the first 15 upregulated genes and the last 15 downregulated genes. We did that to search if there was already information about them in the literature. As you can see, there is already plenty of information about them, and the ones painted in yellow have already been discovered as genes related to breast cancer, and the ones painted in blue have already been discovered as genes related to overweight or feeding problems. And so what we conclude here is that the genes painted in gray right here on the left, they are considered to be new information about the relation between breast cancer and overweight. So we performed the analysis with Enricher, and well, it is well known that overweight is a risk factor for many diseases related to inflammation and the immune system. As you can see here, we have type 1 diabetes, asthma, and some other conditions related to the immune system, right? However, not only these findings were interesting. As you can see here on line 20, we have the leishmaniasis pathway, which could mean that people with overweight and breast cancer have more predisposition to contract parasites from Leishmania gender. And another interesting finding is related to HIV infection on line 11 here. The p-value is not that low, but it could mean that people from this study may have more or less resistance to HIV infection. And then we performed the analysis with network analysts, and we found some other near correlated pathways that are interesting here.
Here you can see the two networks generated from Gene Mania with 20 upregulated genes and 20 downregulated ones from the list of 197 differentially expressed genes. So now focusing on the network created with the 20 upregulated genes, the program has shown that between 20, 13 genes had one or multiple networks that relates one from another, and has also predicted 20 related genes that have connections to 13. In addition, these 20 related genes weren't even present in our differentially expressed gene list, just as the information GAD2, glutamate decarboxylase 2 gene, or the green arrow in the presentation presents the highest number of networks for its relationship with GAD1. On the other hand, HeLa-E, the major histocompatibility complex class IE, or the one pointed with the blue arrow, having connections with MAPK8 gene, TAPBP gene, C19 or F66 gene, B2M gene, and ISG15 gene, is a predicted gene which is linked to the largest number of related genes. Altogether, the map presents 33 genes and a composite of all the networks which best connects related genes between those links are mostly subject to co-expression and physical interactions by 58 and 20%. Now this slide shows map which includes all the 197 differentially expressed genes made through network analysis through minimizing the network, 227 nodes, 555 edges, 122 seeds compose the network analysis. The image shows the network in which upregulated genes, downregulated genes, and added genes are presented in red, mint, and gray nodes. Between all 227 nodes, UBC, ubiquitin C gene has the highest degree and betweenness. In fact, this is one of the genes added by the program. Between the upregulated ones, ESR1 gene has the highest degree and betweenness. On the other hand, an interesting fact is that there are numbers of upregulated genes of overweighted women with breast cancer tied with some virus infection. 9 genes were related with Epstein-Barr virus infection, another 9 of them with herpes, and 5 of them with infection of influenza A and HTLV-I. This proves that there is a possible susceptibility of overweighted women with breast cancer being vulnerable to numbers of such virus infection.
Good night, teacher. Good night, everyone. I'm Fabio, and I'm gonna show to you what we conclude about our research. So, after combining and analyzing the research made on literature with our results found in databases, we cannot be 100% sure that there is a strong connection with breast cancer development and overweight. Once we collect just a few pieces of information about the genes, pathways, and metabolites that are correlated with the disease in the overweight state, considering that it's a super complex disease and the overweight state has a major influence on the human metabolism and body regulation in general. But there are significantly upregulated genes expressed in overweight women that are related to the mechanism of breast cancer disease. So we can assume that there is a relationship between the development of breast cancer and overweight, even though we don't know exactly how strong and how it works. Besides that, we discover a new consequence of this relationship, the increase of susceptibility to acquire viral and leishmaniasis infections, based on the fact that there are upregulated and overlapped genes expressed in women with breast cancer and overweight, which are related with the mechanism of viral disease and leishmaniasis, as it was shown by my colleagues. That's it. Thank you very much.